Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is also visible. Welcome all of you for Human Health and Diseases major concept class. So are you all ready for the class? We are going to pick some topics from this chapter and we are going to thoroughly understand and learn it with me. Okay, ready? All of you quickly let me know if I am audible, visible and also if we are live yet. Okay, I will also check. Yes, we are live, right? Awesome. Okay, now I can see all of your chats. Very nice. Yes, so ready? Audio is visible and video is audible. Very nice. Okay, let's start the class. Okay, so students, we are going to do human health and diseases today. So human health and diseases is one of the important chapters because it has a lot of concepts and it is easy to understand the concepts. So this class, it's not going to be like a normal where I start with common diseases. I'm going to pick some topics of this class and I'm going to make you understand, okay? And it is going to be a short class. It's not going to be a very long class because we are not going to do the entire chapter. We are going to do it in at least 2 parts, okay? Where today I will make you understand about immunity, about some major topics, and later the next part we will be doing. Okay, clear? Yes, all of you. I hope all of you have marked your attendance as well. Okay, like the video quickly and we will start the class. So I want all of you to have 100% concentration. Write only short notes but don't write a lot, okay, because this notes will be shared on Vedantu NEET English Telegram. Okay, ready? Hello, hello, good morning, good morning. Okay, so yes, so students, we are going to start with first, let's respect the chapter name. No, what is health? So health, usually for most of the people it is I am physically fit, I am healthy. Okay, for some people, okay, today I have drank tender coconut water or I have drank, you know, fruit juice, so I am healthy. That is not actually health, right? It has a physical, you should be physically, mentally, socially well with each other, and it is not just mere lack of diseases. Just because you do not have a disease at this moment, but you're physically, mentally very disturbed or in a very bad state, that means you're not healthy. Okay, that means you're not healthy. So health basically means a situation where you are physically, okay, physically, mentally and socially, okay, well. That means your well-being has to be in all these 3 places, that is, be it physical, be it social and be it mentally. If you're doing at all these three states, if you're well, that means you're a healthy person. Okay, now the next heading of the topic is what? Disease, human health and disease. Now what is disease? From the term itself, students, diseases, right? From the term itself you understand one thing, dis-ease, that means you are in a dis-ease, that means you're not in a comfortable stage, right? It's an uncomfortable stage, uncomfortable stage, right? Because you're not feeling at ease, you're not feeling relaxed, because of which we call dis-ease, means an uncomfortable stage or a stage where you are attacked by a pathogen, okay, where a pathogen, a disease causing agent has affected you and because of which you have got a disease. Okay, how does a disease happen? Because of a pathogen, right? A pathogen is someone who has the ability to cause a disease. Clear to all of you? Yes, not in comfort, not in comfort. So this is what the chapter is talking about. Health means what? Physical, mental and social well-being. Disease means dis-ease. You're not at ease, you're not comfortable, and that uncomfortable stage has come to us because of a pathogen who is capable of causing us disease. Okay, causing disease. Clear to all of you? Introduction is clear. Then terms in the chapter, okay, human health and disease. Is this clear? Are you all healthy? Tell me, are you all healthy now? I want answers. Are you all healthy right now? Hello. Yes, hello GMAs. Very nice. Tell me all of you quickly, are you healthy now? Thank you. Okay. Now students, we are going to learn some more important topics. So I hope introduction is clear. Now this chapter has different categorization, right? in your NCERT, we are going to start with common diseases in man. And you learn about typhoid, pneumonia, amoebiasis, all this, okay, common diseases. Then we slowly go to a topic called immunity. Okay, in immunity you will study innate immunity, acquired immunity, you learn about antibodies, you learn about different immunoglobulins, etc. Okay, then we go to different types like AIDS. Okay, you talk about AIDS, you talk about cancer. Okay, then you come to immunization, vaccination, all that. Okay, immunization, and then you go to drug abuse. Okay, drug abuse and alcohol abuse. Okay, alcohol abuse. So this is how the chapter goes. It's a long chapter. So we have already done RNA series of this chapter, and many of them asked me for NCERT line by line. I told no, we will not do NCERT line by line because there's so much. Okay, you do not have to go through that much. You can just learn this chapter this way. So today, students, we are going to pick the main topic. Is going to be immunity. I'm going to pick immunity topic and I'm going to make you understand that topic. Okay, because that topic has a good chance of many questions coming from this chapter, especially from that one topic. So I'm going to teach you that topic. Ready, all of you? Okay, now students, immunity can be divided two types: non-specific and specific. That means when a pathogen is coming, a generalized attack is done from our body. That is, does not matter, like virus is affecting fungi, it's affecting bacteria, helminths are affecting, not an issue for us. It is a generalized, it's not specific. Okay, it's not specific. Now I'm not picking upon one student, right? I'm teaching all of you the same way. Imagine I'm picking upon only one student, or I'm only focusing on one student. That means what? I am very specific that I need to teach only this one student, right? So now same thing here. What is going to happen here? It is where this whole thing is not seen, huh? Guys, whole screen is not seen. One second, let me just check. It's seen here. Is it the... Is the whole screen visible to all of you? Okay, it's not visible, no? Wait, I have to fix that now. Okay, just give me 1 second, guys. 1 second, okay? Not seen, no? Yeah, 1 second, because I will write there and you will not be able to see it. Okay, I'll just drop a message. They will just come and fix it, okay? Yes, right side is not visible, no? 1 second. I'll just drop a message. Okay, done. Okay, they'll fix it, so we'll continue the class. Okay, yes. Okay, yeah, right side is not visible. It is live, NEET UG 2024. Okay, now students, see, immunity, as I told you, it can be specific and it can be non-specific. That means a generalized way of dealing with it or a specialized way of dealing with it. Okay, so there are some terminologies which I'm going to teach you, from where I saw some questions are also being come. Okay, so here let's talk about your immunity. Now students, what is immunity? I think this word has become very common to us during the COVID era, where everyone started talking about immunity, telling that, oh my god, they have strong immunity, we do not have strong immunity, because of which we might get affected. We might get affected by this virus, right? All of you have heard about the word immunity. That means the ability of our body to fight against the disease causing agents, be it anything, be it virus, be it fungi, be it bacteria, be it helminths, anything, protozoans. So the ability of our body to fight against a disease causing, that is our immunity, right? So that immunity again can be categorized into first line, second line, third line of defense. Okay, now students, immunity word is clear. Do I write it here? Okay, ability of the host to fight against disease causing organisms. okay, disease causing organism. You can also call it an antigen, right? So this is immunity. Now I am telling that immunity can be divided into two categories, okay? Immunity can be divided into two categories. First one is non-specific, okay, non-specific, and another one is specific, okay, yes. Yes, they are fixing it, okay, 1 second. Yeah, fine, fine. This A is missing, little more. It's fine. This is fine. This is fine. Yeah. Yeah. Okay, now it's visible, right? Everything is visible? Yes, perfect, perfect. Okay. Now, look here, non-specific and specific. That means what? As I already mentioned you, non-specific means it's a very generalized way of dealing with something, right? Very generalized way of dealing with something. Now look here, this is going to be a generalized way of dealing with. Now all of you tell me in the chat box, if it is a generalized way, are they using antibodies? All of you tell me, if it is a generalized way of dealing with something, that means an antigen, a problematic fellow has come, okay, and they are dealing it very generalized, okay, we are not specific anything, but are we going to use antibodies? Yes or no question in the chat, okay, are we going to use antibodies? By then I am going to write this. Specific means obviously specialized, okay, specialized kind of treatment, okay. All of you, advance happy Diwali, right? Tomorrow I think all of you will be celebrating, and some of you might be celebrating on 1st of November, right? So yes, advance happy Diwali to all of you. I hope all of you are safe. Do not burst crackers, which are very dangerous for you, and do not do any kind of, you know, circus with the crackers that you hold it in the hand and you want to be Bahubali. Don't do any of that. Do it in a proper way. Even if you want to burst crackers, do it in a proper way, and make sure that you take care of the animals, okay? Don't tie it on their tail, you know, don't put colors on their face, don't do all that kind of things. You can have fun without harming anything else around you, right? Yes, okay? So yes, all of you look here. So non-specific means generalized. So here students, we are going to fight, okay, we are going to fight the antigens or fight the, you know, pathogens without the help of antibodies, okay, fight the pathogens without the help of antibodies, without antibodies. Clear to all of you? Yes, without antibodies. But since it is specific, obviously ma'am, I am going to use antibodies. So here we are going to fight the pathogens with the help of antibodies. Clear to all of you? This is important. Question can come. In non-specific, do they have antibodies? Very simple question, but the students will be like, oh yeah, no, everything is immunity, so obviously antibodies should be there. No, okay, it is not necessary. Even though we talk about immunity, we keep talking about, you know, immunity as a whole. We should understand that when we talk about immunity, we are stressing on specific immunity, right? Who is going to fight exactly? Okay, this fellow has come. I know exactly how to tackle him. Many people have come. Everyone I will tackle the same way. It does not work that way, right? Now, all of your understanding capabilities is different. Some of you might be slow learners. Some of you might be fast learners. Some of you grasp very fast and forget very fast. Some of you grasp very slow, remember it for longer time. So, all of you, the way you learn have to be different. Understood? Yes? So, this is the thing. So, here, so much we have understood immunity, non-specific and specific. Now we are going to learn something called as first line of, you know, defense, second line of defense and third line of defense. So students, in this non-specific, there can be two categories, okay, further divided into two categories, which is called your external, okay, external and internal, okay, external and internal. Now from the terms, do not be like, oh my god, what is external and internal? External means what? Your skin is there. right? Your entire body is covered of skin. Your skin becomes the first line of defense, right? If anything is entering, first it has to fight with your skin. It has to fight with your stratified squamous epithelium. It has to fight with the layers of the skin. After that, whom do we have? We have mucous layers, right? Wherein all in our body mucous membranes are covered, mucous layers are covered. All those portions also, they are going to fight, right? Be it your respiratory tract, be it your urinogenital tract, all these places also, whenever something is entering, they are going to fight against us. So far good? Clear or not? Wow, wow. What an all names, students, for YouTube. Lacoperon, it seems. Okay, lacoperon, oxytocin, pastry. Why, students, your parents haven't kept, such beautiful names they would have kept for you. So much they would have researched to keep your, Gen Z names, and you people put lacoperon, translation, transcription as your names. Okay, full parents, that time is wasted. Yes, so this is clear. External and internal. So when I'm talking about your immunity as a whole, you would have noticed that in your textbook they have given two types of immunity. What are they? What are they? Students, if you do not take anything from this class, okay, because you're talking to your friends, let me tell you, it is going to be a loss for you, because I'm doing a revision, okay, and I'm picking only one or two topics and I'm going to teach it slowly, making you understand every single word, okay. So if you're not going to concentrate and if you're not going to have your presence of mind here, then the whole class is going to be useless, okay. So listen to me carefully. Love towards bio, too much love it has become, okay. Now tell me, lacoperon, who is an inducer? And students, why haven't you all liked the video? You're learning something, no? So you should immediately like the video. That's how I know that you're also enjoying the class. How much I am enjoying teaching you, you should also enjoy learning with me. So all of you like the video quickly. Immunity is two types, students. Innate immunity, okay, and acquired immunity. Innate immunity and acquired immunity. So under innate immunity, they have given you 4 barriers. Barriers means what? Something that is going to block something, right? Same thing, skin is your barrier, mucous membrane is a barrier, same way you have physiological barrier, cellular barriers, cytokine barriers, correct? So all this is going to be spoken under your innate immunity. Now if I talk about innate immunity, when I talk about this whole thing, okay, non-specific, innate immunity is non-specific. It is there from birth, okay. By birth you have it. Now a baby is born, okay, let's say a baby is born and we'll name the baby as translation. Now that baby is not exposed to any pathogens which is outside. So the body also does not have any memory, any response to any of these pathogens, because they have not encountered it, right? But what is acquired immunity? These are the diseases, or these are the, problems that we are going to acquire later in our lives, right? Let's say cancer, okay? Let's say AIDS. All this happened later in the life. So until it is, transferred from the mother to the baby, that is a different story, but I'm telling, okay, it is usually in the lifetime. Okay, acquired during the lifetime, okay, during the lifetime. So this is the most important thing, okay, most important thing. What happened? What happened? Okay, yes. Clear? Fine? Works? Okay. Now under innate immunity, students, when we talk about your external and internal also, we will understand that those are your first and second line of defense, okay. They are going to be the first line of defense. Who is going to be the first line of defense? Your skin, right, your physiological barriers, be it your tears, saliva, all these, okay, ear wax, okay, cerumen, all these are going to be your first line of defense, right? Then you have second line of defense. Second line of defense, that means imagine after, somehow they escaped the first line of defense. They escaped your skin, they escaped your saliva, tears, and they came in contact with your blood. So once it enters into your body, you have WBCs, you have NK cells, you have macrophages, all these also will be ready to fight, telling that, okay, I'm going to fight with you. Then you have lymphoid organs, all your secondary, primary lymphoid organs, all these are going to fight, okay. So you have first line of defense and second line of defense, and under acquired you will have third line of defense. Is this clear to all of you? Yes or no? Third line of defense. Clear? Yes, quickly answer in the chat box. Students, I don't want all of you to talk about someone else. Please listen to the class, okay? Do not waste your time and do not waste other people's time as well. Got it? Yes, all Kanjus in the class. Okay, no problem. Now tell me, so far good? I want thumbs up in the chat box if you understood so far. What did I tell you? I spoke about immunity to all of you. I spoke about non-specific, specific. Non-specific is generalized. Specific is specialized. Okay. Generalized, that means we fight pathogens without the help of antibodies because it's non-specific. Antibodies are going to be specific, right? So without the antibody. And there are two types, external and internal. External means what? Like your skin, like your mucous membrane, physiological barriers. Those are all going to come in external. Like an internal, what we have, again, when it enters into our body, you also have WBCs, macrophages, NK cells to fight them, okay? Then here if you go, students, immunity in your textbook is categorized into innate and acquired immunity. Innate is something that we have by birth. Acquired is during our lifetime we get it, okay? Now again, both under innate immunity you will have your first line of defense and second line of defense, and under acquired you will have your third line of defense. Is this clear? These terminologies, please make it clear, okay? Now, next thing, okay, all these notes will be available on the Telegram Biology by Gopika, and even if you want you can follow me on the Instagram as well, again Biology by Gopika, okay? Gopika Biology on Instagram, but even if you type Biology by Gopika, it's going to come, okay? So all the notes will be available on Vedantu NEET English Telegram as well. That is our main Telegram where we share all the content of all the teachers. Clear to all of you? Perfect. So far good? Yes, very nice. Now let's start with skin, okay? So students, under innate immunity, if you see, there are barriers which are given, right? So I'm going to write the barriers. There are barriers which are given under your innate immunity in your textbook, and the first barrier, what do they call it as? Physical barrier, okay? The first barrier they talk about is your physical barrier. Now under physical barrier, what is the first thing? Normally, what is going to protect you? Your skin, right? Your skin. Now tell me the layers of the skin. Okay, so skin is going to be the first protective layer, and the skin has a layer. What is that layer called as? It is keratinized. Okay, keratinized, stratified, squamous epithelium. Yes or no? I taught you this in, animal tissues. Squamous epithelium. Okay. Now, keratinized. Because the word keratinized you saw, you'll be like, ma'am, but our skin is so soft. How is that possible? Because on our skin, we have oil glands. Okay. We have oil glands that is going to do the lubrication or going to keep our skin very soft. Clear? Soft skin is because of the presence of oil glands, sweat glands who are making our skin soft. Even though the skin has a keratinized stratified squamous epithelium, our skin is still soft. Clear? Very good, Tanuja. Very good. So this is the first layer of defense where skin is going to do the protection. Clear to all of you? Very nice. Now, our skin has some bacteria, okay? Staphylococcus epidermidis. That bacteria, you do not have to remember it, but remember that there are bacteria present on our skin which is going to maintain the pH in the skin. Now, ma'am, why are you telling us this about, why are we talking about this? It's because students understand the pH of the skin is very important, because if the pH is acidic, that means we will get less infections on our body, okay? So, the skin will try to avoid any kind of infection because of the acidic pH. Is this clear? Can you all remember all this? Quickly let me know in the chats. Yes, the name is Staphylococcus epidermidis. That is the bacteria's name, okay? If you want, you can remember, otherwise no problem at all. Okay, very nice. What is that bacteria going to do? It is going to make sure, okay, it is going to make sure that the pH of our skin is maintained, okay? So, our skin pH is a little acidic. Okay, skin pH is a little acidic, and this acidic pH actually makes sure that we are having less of infection. So, it does not allow, okay, infections to bloom. So, it minimizes, we can say it minimizes chances of infection. chances of infection. Clear? Clear? You might have a question. No, ma'am, just by having a layer, how are they going to do the protection? They have to have this. Clear? Okay, done, done, done. Now, this physical barrier is clear that you have skin. Now, you also have something called as your mucus, okay, mucus lining. That is, wherever the mucus membrane is there is going to be mucus lining. Now, you tell me which are the most common openings in our body through which a pathogen can enter. First is going to be my nostrils, right? Because I'm going to inhale the air. I'm going to inhale whatever is there in the atmosphere. That is going to be first way of entering into my body, right? My pathogen can enter through my respiratory tract. Then it can enter through my urinogenital tract. Then it can enter to my digestive tract. That means through the mouth, through the nostrils and through your openings, okay? In the openings in your reproductive urinogenital tracts can let pathogens enter inside. So these three are the openings through which the pathogens are in direct contact with us, right? They can easily enter us. So because of which these places we have mucous membranes, okay? What do we have? Mucous membranes. So here we are going to write that mucus, okay? Mucus is present, that is because of the presence of mucous membrane. Okay, mucous membrane. And where are they present, students? Tell me, all of you in the chats. Chat is not at all moving. Okay, all of you have to be more energetic in the class. Yes, very good. Marjorie has told respiratory tract. What about others? What about others? Yes, very good. So, mucous membrane is going to be present, okay, in your respiratory tract. Okay, respiratory tract. In your gastrointestinal tract, okay, and urinogenital. Correct? Agreed, all of you? These are the places where the mucus is going to be there, and what are they going to do? They are going to make sure to filter, right? Filter the dust particles. Okay, filter the dust particles, also filter and protect us from any kind of pathogen that has entered. Okay, pathogen that has entered. Clear? Thumbs up? Yes, very good. Urinogenital, gastrointestinal and your respiratory tract. These are the regions where your mucous membranes are there, where mucus will be secreted, and this mucus make sure that sometimes all of you would have heard, no, I do not know it in your language, but sometimes when we are just sitting, okay, suddenly you feel like a mucus is rushing out, right? You feel that mucus is coming out suddenly and you'll be like, what happened? I do not even have a cough. What is happening? Basically, they're lining, you have your ciliated membranes, no? They're pushing the mucus up because it could have had some mix of something, okay? It could have had some dust particles which is irritating your body. So because of which it will be pushed out. Clear? That's why you go suddenly and spit it out. Perfect? Yes, very good, okay, fine, understood. So these are going to entrap the foreign particles, okay, instead of pathogen, I'm going to write it traps, okay, it traps the foreign particles, okay, foreign particles. Done. So these are two physical barriers that we are going to talk about. Okay, we finished talking about it. So these are going to be your physical barriers, also your first line of defense. Okay, first line of defense. Physical barriers. Yes, mucus as well as your skin. Done? Okay. Okay, done. Exactly. No blue hearts, red hearts. Or fire emojis, right? Okay, next one is going to be physiological barriers, which is again mentioned in your NCERT. Physiological, okay, barriers. Physiological barriers. Now, what is physiological barriers? The barriers, okay, which are present, like imagine by somehow it has entered, the food or the dust has entered. Okay, now what do? Now, example, you have first your tears. Okay, the tears are going to have lysozymes which are going to make sure to damage the gram-positive bacteria or destroy the gram-positive bacteria. Then in your saliva also there is lysozyme. Same way HCl in your stomach, which is acidic, okay, and they are very potent, you know, people to fight your Salmonella typhi, that is your typhoid causing bacteria. All this HCl, saliva, you know, earwax, your cerumen, all these people are going to fight against the pathogens which are entering. Okay, yes, very good, very good. So physiological barriers are going to include your saliva, okay, tears, earwax, cerumen, okay, then saliva, tears, earwax. I think these three are given to all of you. Did we miss out on something? Yes, HCl. Okay, HCl. So students, these two people, who your first two people are going to have lysozyme. Okay, lyso. These are going to make sure to protect us from any kind of problems. Okay, actually lysozyme was first. Okay, first what happened, students, Alexander Fleming, he was working on something, a plate, okay, like plate matlab he was doing with the, he was working on different types of bacteria in the lab, and suddenly he happened to sneeze, okay, and he was like, okay, this plate, this petri plate with all the culture is going to be wasted, okay, but later he noticed that in that plate the bacteria did not grow, because he understood that, oh my god, this saliva which or the sneeze, okay, it had something that was protecting me from bacteria. So that was actually you know, by fluke we found out that Alexander Fleming found out the word lysozyme, or he found lysozyme, that is something that can protect us from pathogens. Okay, clear? So saliva, tears have lysozymes which are going to protect us from any kind of foreign particles. Then you have earwax and HCl, and this HCl is very potent, that means it is very strong acid which is going to fight against your Salmonella typhi. Salmonella typhi. Okay, clear? All of you, so far good? Physiological barriers, so far good? Yes? Rhinovirus. Okay, good. Yes, secretions from the skin, mucus, glands. Okay, yes, very nice. Now students, even your sweat, okay, sweat also is one way of protecting itself, because sweat is going to remove out the dust particle. We have learned this in your excretion as well. They are also going to be accessory, okay, organs which are going to give out some nitrogenous waste. It's not high amounts, but some of it will be given. Now in women vagina, all of you know that women vagina also, the pH is maintained. Why the pH have to be maintained? Acidic pH is maintained in the vagina of women, so that because it is also exposed, right, there is high chances that we catch an infection. That is why you shouldn't actually technically use all these, you know, different brands which are selling, you know, vaginal washing, all that, you know, nowadays ads are too much, but it is not necessary, because our vagina itself knows how to regulate the pH with the help of bacteria. Okay, so that bacteria will do the job of regulating, the good bacteria which will do the job of regulating the pH, acidic pH in the vagina, because of which we will not be exposed to any infections. Okay, but what happens is, when we use soap, when we use shampoo by mistake, when we use our body wash or any kind of V-wash, other things, the pH will be disturbed. When the pH is disturbed, it becomes alkaline, or it changes, the acidity changes. That means you are more prone to infection, and the brands that sells it does not tell this. Okay, they will be like, your pH will be maintained. How can something's pH be maintained? Their soap. Okay, all of you have studied in chemistry, no, they are alkaline, soaps are there. So obviously they are going to wash out and they are going to make it diluted. The acidic pH will be changed. Clear to all of you? Yes or no? Clear? Yes. Soaps, yeah, soaps. Perfect, perfect. Okay. Yes, the bacteria's name is actually Lactobacillus acidophilus. Okay, Lactobacillus acidophilus is the bacteria which actually maintains the vaginal pH. If you want, I can write one line here. Okay, if you want, I'll write a line here. So, vaginal pH have to be acidic. Okay, why it has to be acidic? So that it can minimize, okay, it can minimize the chances of infection. Okay, infection. And the good bacteria which is going to do that, it's going to be your Lactobacillus acidophilus. Lactobacillus acidophilus. Okay, this is going to maintain the pH. Clear? Yes, it is going to maintain the acidic pH. Clear? Okay, it's like one ASMR video, no? Pen sound is coming. Yes, minimize infection. Done. Physiological barrier is clear. Any secretions from your skin, mucus, your saliva, tears, earwax, all of this also comes as your first line of defense, because these people have, our body is made in such a way that wherever there is an opening, wherever there is chance for exposure to a pathogen, our body has already given a coating for it, be it mucus, be it tears, be it saliva, be it earwax. So by mistake it enters also, it goes to the ear will be like, I have the earwax, okay, I'm going to fight with it. Now if it enters into your eyes also, tears are going to fight. That's why the moment something enters into your eyes, you start tearing up. Why? Because your body is trying to flush it out. Okay, it is going to flush it out, and that is why your tears starts coming immediately. Okay, done. Yes, clear? Okay, clear. Okay, fine. Now we will go to internal defense. So students, now we have understood both the external defense, right? Here, okay, here external defense we have understood, your skin was there, mucus was there, and also your physiological barriers are there. Now we have to go to internal defense. Now what is internal defense? That means your cellular barriers in other terms. Okay, in your textbook only it is given, no? Internal, okay, internal barriers. Now that we can also say as cellular barriers. Okay, cellular barriers. Now imagine pathogen is too smart, okay, over smart pathogen has entered inside your blood vessels. Okay, even though we had so many external barriers, imagine pathogen has entered into your blood vessel. Now what is going to happen? It has entered into your blood vessels. Now we have to fight against it, right? We have to fight against it. That's the evil person who has entered. Now for that also our cells are going to be there. Who are that cells? Your WBC is going to be there, right? Your WBCs, macrophages, NK cell, that is natural killer cells, all these people are also present inside. So just because the pathogen entered inside does not mean we are going to get affected by it, because we have cells like your WBC, macrophages, natural killer cells, you have your neutrophils, okay, neutrophils, which is also termed as PMNL, okay, polymorphonuclear leukocytes, you can tell, because again WBC, no, so neutrophils, okay, or else you can also use the word cells. So PMNL or PMNC, that is polymorphonuclear cells, okay. These cells are also present inside. So imagine this pathogen, after surpassing all the external things, it has entered into our body. Now how it would have entered into the body? Let's say Smile official is one student here, okay. Smile was running, okay, playing with his friends, and suddenly he got a scratch from an iron nail, or he got a scratch from something, some metallic thing. Now what happened? Immediately the cut has happened. So the pathogen would have entered through that cut, right? It would have directly come in contact with the blood and it would have entered. So it did not get enough time to fight, or our body did not get enough time to fight with or push it out through the physical barriers, because there was a cut and immediately it came in contact with the blood. So it would have entered inside. Now once it enters inside also, there is no high chances that okay, immediately you will get an infection. No, you will have WBCs, macrophages, NK cells and your neutrophils who will be ready to fight inside your body. Now all of you remember WBCs, I told something. Okay, yeah, Smile is crying. Now students, remember this WBCs have a very extreme one thing that they can do. That is, let's say this is your capillaries. Okay, these are your capillaries. I can't see only, students, 1 second. Okay, these are your capillaries. Now what is going to happen? This WBCs have the ability to squeeze out. Okay, WBCs have the ability to squeeze out of the capillaries and move towards the infected cells because of their amoeboid movement, which we studied yesterday in locomotion and movement. That sorry, 1 second. Okay. Yes, students, all of you like the video. Quickly, jaldi, we should have now 100 likes faster. Yes, quickly, okay, all of you are enjoying, so you should definitely mark your attendance as well. Yes or no? Yes or no? Yes, cry. You people are funny. Okay, now students, look here. This WBCs have a special skill. What is the special skill that they have? That is, they will be able to squeeze out of the pores of the capillaries, through the pores of the capillaries towards the infected cell. So let's say here the infected cell is there. These will be able to squeeze out. What is this process called as? I have taught you this in body fluids and circulation. Who is going to answer this? Who is going to answer this? Yes, we are going to come to that, Harish. We are going to come to cytokine barriers. That time I'll teach you this, okay? Right now, let's learn what we are learning right now. Quickly tell me, what is the process name I told all of you? That when WBCs are squeezing out, okay, squeezing out, there's some word that we use. It's called diapedesis. It is called what? It is called diapedesis. Okay, a situation where, a condition where your WBCs will squeeze out. Okay, it will squeeze out of the capillaries and move towards the infected cells. Okay. Done. Clear? So there you have your cellular barriers. Now we are very good, diapedesis. Now we are going to go to a next condition, that is going to be your cytokine barriers. Okay, cytokine barriers. So cytokine barriers, students, here we have to learn few things. Okay, first we will draw two cells. Okay, I'm going to make you understand with the help of a cell itself. Now let's say two cells are there. Okay, this is our first cell. Who wants to be cell 1? Who wants to be cell 2? Okay, no one should be. Now we are going to just understand. This is our cell 1, cell 2. Okay, done. Okay, now say, students, this is our cell A. Okay, cell A, and this is our cell B. Now what has happened? This cell, the cell A, got affected by a virus. Okay, now I am drawing a bacteriophage. It got affected by a virus. Now if it got affected, okay, it got affected by a virus, what is it going to do? It is going to immediately add its genetic material, right? Okay, 1 second, students. Okay, it is going to add its genetic material into the cell and go and bind with our genetic material and start forming our viral proteins, right? That is what these viruses are going to do. As soon as they attack the cell, they are going to release their genetic materials. Okay, release their genetic material into the cell, and that genetic material is going to bind with the host cell, okay, host DNA, and they are going to start forming the viral proteins. This is generally what happens. Yes or no? Now there is a mechanism in our body, there is a barrier in our body which is called the cytokine barrier. Okay, barrier which is called the cytokine barrier. Now this cytokine barrier, students, understand there is some word called interferons. I'll tell you what exactly happens. This fellow, that is cell A, already understood that I am going to die because virus has affected me and I cannot do anything about it, right? Because they are non-living. The moment it comes in contact with the host, it becomes active. It will start doing its replication, translation, transcription. All that we can do is stop their translation from happening. Now, what is exactly going to happen? Cell A already knows that I am going to die. So, cell A will release something called as interferons, okay? Cell A will release something called as interferons. I'll write it with a different color. Interferons. Interferons. Now, this interferons are such that it's a protein which is going to come and bind with the next cells, okay? Next cells also many are there. It is going to, these cells are going to have receptors which are going to accept this interferons, okay? Accept this interferons. They are going to take this interferons and suddenly realize that, okay, cell A was in problem, I shouldn't be in problem. So, they are going to make antiviral proteins, okay? They are going to make antiviral proteins and protect them from getting affected by a virus. Clear to all of you? Clear to all of you? Yes or no? So, there are interferons which are released that will go and bind with the receptor of the next cell or bind with the whole cell, and that cell is going to realize that, okay, cell A was in problem, I shouldn't be in problem, I got the signal, so I am going to form antiviral proteins that protect myself from virus. Simple as that. Yes or no? Protect myself from virus. Protect from viral infection. That is why, students, in our body also, if you have noticed, first there is going to be, if you draw a graph of how our virus affects our body, you will always notice that there is going to be a peak phase, because body is still getting used to this, no, it does not know that this has happened. So, first it is going to go have an exponential growth. After some time, it is going to go to a lag phase. Then it is going to go into a stationary phase. Why is this happening? It is happening because of the same thing. Because body will start, the other cells will start sending signals, start sending interferons. That interferons will make sure that the next cell forms antiviral protein. This will keep continuing. By then, our body also will start having, will fight this virus, and our body will go to a lag phase. That means the virus will no more be able to have an exponential growth. Clear? All of you? Yes, my voice is cracking. No, again? I don't know what is wrong with my voice now. Clear? Yes. Oh my God. So away from the target. All of you, this is super clear. Students, we can also use the word AVP, okay, this one, antiviral protein, AVP, okay, some places short forms are written, so you understand AVP full form is antiviral protein. Done, done. All of you, happy, clear, remember everything? Okay, what has happened? This is a kind of cytokine barriers. Have I written it somewhere? No, right? So, this is the type of cytokine barriers which is going to fall under your innate immunity. So, we have learned all the 4 barriers of innate immunity. We started with physical barriers, then we went to physiological barriers, then we went to your cellular barriers, and at last we learned cytokine barriers, okay. So, here interferons play a very important role, which is going to signal the other cells, telling that please make antiviral proteins because I got affected by virus. Now, you shouldn't get affected by virus, and that is the only reason why in our body, if we have a viral infection, it starts exponentially, then it goes to a lag phase. Done, done. Is this clear? Okay, very nice, very nice. Any doubts? Any doubts so far? If you have doubts, please ask me. All these notes will be available on Telegram, so don't take tension of it, okay? Students, shall I write here the barriers down? Okay, shall I write the barriers down? Okay, I'll write it in a box so that you know. Some students, what happen, no, they blindly study the notes and they do not even open NCERT and check once. So, we have to write everything once again and again for them. Okay, so here I am just going to write your innate immunity. Okay, innate immunity. They have barriers. Okay, they have barriers starting with your physical barrier. Okay, physical barrier, physiological, cellular, and cytokine. Cytokine barriers. Okay, so I've written it in a box here, so when you get the notes it will be clear. Clear? Okay. Now students, we are going to move on to acquired immunity. Now innate immunity is clear, immunity as a whole is clear. Now we are going to learn the third line of defense. Okay, acquired immunity. That means, let's say I do not have any exposure to this. For the first time, I am exposing to some pathogens, that will become my primary response. Then when this pathogen comes second time, my body will be like, hey, I know you, I remember you, I have memory of it, okay. They have memory cells. So I have memory of this, that becomes your secondary immune response. All this I think all of you know, right? All of you know, right? So students, we are going to talk about acquired, okay, acquired immunity. Acquired immunity. What is acquired immunity? Something that we have, okay, we have came across this pathogen for during our lifetime, okay, and to that when our body responds, that becomes our acquired immunity. Done to all of you? Yes, very good. Now the most important part about acquired immunity is pathogen specific. Okay, they are pathogen specific immunity. That means they are very specific. They are not going to attack generalized way, they are going to attack in a specific manner. But innate immunity is a non-specific and they are going to attack in a generalized manner. Clear? Okay, pathogen specific, and students, they will also have memory. Okay, they will have memory of the pathogen that they encountered. And when it is going to happen for the first time, let's say Tanuja first time ever came across a virus, okay, that means what, her body is going to do a primary response, that is first ever approach, right? First encounter is going to be the primary response, and when it happens the second time, let's say this virus came the second time, it is going to become a secondary response. Clear to all of you? Primary response is going to be your first, okay, body is encountering it for the first time. So shall I write it that way only? Body is encountering for the first time. Clear? Now secondary response is going to be when it is going to affect the second time, okay, when it enters the second time. Clear? Easy peasy. Yes? My God, once I tell something, they'll go off. Run. Okay, very nice. So far good? Any doubts so far? Okay. Yes, and also one more thing add on that I want to tell about acquired immunity students, they will be able to understand, differentiate between self cells and non-self cells. That means they'll be able to understand who are my genuine friends, who are my fake friends, no, like that. So same way here, they will be able to understand the self cells, my own cells, and the cells which have come from outside, okay. So they will be able to differentiate, okay, differentiate self cells from non-self cells. Clear? They will be able to discriminate, differentiate self cells, non-self cells. They will have diversity. They will be able to understand diversity of pathogens are there. Got it? Yes. Okay. Clear? Okay. Something and all you people are doing. Now students, one more topic that you have to learn under this itself is your lymphoid organs. What is it? Lymphoid organs. Okay, so I'm going to write it here only, but the problem is, okay, this I'm just going to write it down, okay. Yes. Now lymphoid organs. All of you tell me, what do you know about them? You'll be like, ma'am, we don't know anything. Guys, after this class, I want all of you to go and practice at least 5-10 questions from this chapter, okay? Don't wait for me to do it in the month of Jan and then practice. We have already reached November. December, January, February will go in a, it will go like one super train, okay? It is going to go that fast. So that's why as soon as you finish this chapter, go and practice at least 5 to 10 questions. I'm not even asking you to do more. 5 to 10 questions, go and practice. Now, where will I go and practice? You can go to NEET Treasure Box. I think the link should be there under this video. Okay, let me check. See, get NEET Preparation Treasure Box. If you click on this link, okay, if you go down all the way and if you go here, you will be able to see NCERT solution chapter wise PYQ PDF will be there. So NEET based, just go to human health and diseases here and get the chapter link, okay? Get the chapter link. See, human health and diseases is here. Go all the way here and get yourself the NEET PDF and practice 10 questions. Got it? Practice 10 questions from it and do not wait for one other Shankranti to come for you to practice questions. Now, lymphoid organs are divided. Okay, now first of all, what is lymphoid organs? Your lymph cells, okay, there are nodes, okay, in our body there are nodes present, especially in women, we have it again underarms, okay, we tell no. So here, near our armpits, near the mammary glands we have it, near our groins also, where our reproductive parts are there. So these areas you have your lymphoid organ, they are all going to do the protection. Now remember that your lymphoid organ, we have two types, okay, two categories we can classify them as: primary lymphoid organ, primary lymphoid organ, primary lymphoid organ, and secondary lymphoid organ, secondary lymphoid organ. Clear to all of you? Primary and secondary lymphoid organ. Now students, lymph nodes are there, okay? They are small, solid nodes are present. These are also going to help us fight the pathogens, okay? Now these nodes are, they are going to be like this bean shaped structures and they will have coming inside, okay, this is going to be the afferent part. Now students, imagine these are your lymphocytes, okay, lymphocytes, cells of the lymph, right, lymphocytes, they are going to fight the pathogens. Lymphocytes are there and they are going to fight the pathogen. Let's say this is the pathogen, okay, this is the pathogen, they are going to fight the pathogen. Clear? So now remember this, everything became pink color, no, too much of pink. Will not be able to differentiate only, I feel. Okay, let's draw it with this color, okay. Okay, this will be kind of kidney shaped, bean shaped, we can tell the lymph. Now to this we have vessels connected. Now inside this you have your lymphocytes, okay. Inside this you have what? Lymphocyte cells. These lymphocytes are going to fight with the pathogens. Clear to all of you? Clear to all of you? Okay, the green color is the pathogen, it is going to fight the pathogen. This is how it happens. Now, primary lymphoid organ, what is it? Secondary lymphoid organ, what is this? That you tell me in the chat. Primary lymphoid organ, two people are there. Who are they? Oh, bone marrow, letters are missing students. Okay, bone marrow and thymus. Bone marrow and thymus. Clear to all of you? Bone marrow and thymus. Now in the bone marrow, what is going to happen? Your B lymphocytes are going to mature and differentiate. In thymus, what is going to happen? B cells, okay, bone marrow, which is present inside your bone, here only the formation of almost everything is happening, RBC, WBC, platelets, everything. But platelets are not actual cells, I have already told you that they are fragments, okay, of your megakaryocyte. Now this bone marrow students, in the bone marrow itself, all these cells are being formed. lymphocytes will be formed, right? Lymphocytes will be formed. Remember granulocyte, agranulocytes, we studied there, neutrophils, basophils, eosinophils, monocytes and lymphocytes. Lymphocytes, B lymphocytes, T lymphocytes, they are going to be formed here, but T lymphocytes are going to differentiate in the thymus. That's how the name also came, okay? B cells and T cells. Even though bursa or Fabricia is mentioned in your B cells, you know, B cells, B stands for that, but it is not true because that is present, the extraction of that happened in the birds. That's a specialized organ in birds, which is not there in humans. So we can just remember this way, that in bone marrow only formation happened, derivation happened, they are there only, differentiation happened, they are there only. Thymus, in the thymus your T cells are going to go, okay? So here B cells, okay, B cells formation and T cell formation is going to happen. Here T cell differentiation is going to happen. Okay, T cells differentiation is going to happen. Oh ho, letters are going missing. Okay, let's hope all the letters land in a correct place. Clear to all of you? Any doubts? Any doubts? Should I repeat anything I told you? Tell me quickly in the chats. Anything that I have to repeat, tell me right now. Yes. Erythropoiesis, that means formation of your RBCs. Now in the bone marrow only, again everything is formed, okay? Be it your RBCs, WBCs, okay, platelets, everything, okay? Now students, remember one thing, when I taught you this, I have to tell you one add-on here. Now I'm just going to, I think here there is no place. So students, when we talk about this, no, always remember your lymphocytes are going to be a part of your specific, okay, immunity. That is acquired immunity. It's going to be proper specific one, because B lymphocytes are going to form antibodies. They are going to be very specific. So when you write WBCs, no, do not write lymphocytes. So better you can write monocytes, macrophages, NK cells and neutrophils, okay. Eosinophils, basophils also you can write, but you shouldn't write by mistake also lymphocytes, because they are specific. Clear? Yes? T lymphocytes, repeat. Okay, so I'm going to teach this in detail right now. Just try to learn the names, okay? Just remember that primary lymphoid organ, bone marrow and thymus is there. In your secondary lymphoid organ, who is going to be there? Secondary lymphoid organ, more people are going to be there, like your tonsils, okay, tonsils, spleen, Peyer's patch, appendix. All these are a part of your secondary lymphoid organ. Just check if I missed something. Okay, all of you in the chat box, just tell me if I missed something quickly. And no, yes, T lymphocytes, I'll repeat here. Don't worry. Don't even worry. Okay, yes, I'm going to repeat it. I'm going to teach that in detail now. Okay, this is clear. Acquired immunity is a pathogen specific immunity. They are going to have memory. Primary response is when it happens for the first time. When our body encounters the pathogen for the first time, it is going to have a primary response. Secondary response, because it has memory, when the pathogen comes again in our body, secondary response is going to act. Okay, now it will be able to differentiate cells and non-self cells. Now students, here lymphoid organ, again it is going to help us to protect our body. Primary lymphoid organ is there, secondary lymphoid organ is there. Lymph nodes are bean-shaped structures. They are going to have lymphocytes, which are cells of the lymph. Now students, primary lymphoid organ, you have bone marrow and thymus, and secondary lymphoid organ, you have tonsils, spleen, okay, Peyer's patch, appendix. Clear? Okay. Inert immunity, there is no inert immunity. Okay, now students, I will talk about your primary lymphoid organ, which is important for you to know. Okay, primary, see how much is there in immunity alone. So if you do not study every day, you are going to forget all of this. Okay, primary lymphoid organ. Primary lymphoid organ. What I told you, two things that we have to study. First one is going to be your bone marrow. Bone marrow. Now students, bone marrow, you have to remember that your maturation and formation of your B cells is going to happen here. What is going to happen? Origin, okay, origin of B cells and T cells is going to happen in your bone marrow. Clear? But maturation, okay, maturation of only B cells is going to happen. Okay, maturation of B cells is only going to happen here. Clear to all of you? Many students get confused. They write origin and maturation of T cells and B cells in bone marrow. Wrong, absolutely wrong. Bone marrow, remember, B for B cells, B for bone marrow, okay. Origin of both is happening, okay, B cells and T cells is happening because of the fact that RBC, WBC, platelets, everything is formed in bone marrow. Then why only this, T cells will go somewhere, right? So, bone marrow, origin of B cell and T cell is happening, but maturation of only B cell is happening, okay. And this B cell only is going to form our antibodies in the future, right? It is going to form the antibodies. Okay, it is going to form the antibodies who are going to fight the antigen, which are the foreign particles. Now, the second thing is going to be what, students? Thymus. Now, thymus is present in the mediastinum, right? Between, okay, just near the heart. Clear? Just near the heart, your thymus is going to be present. Again, you can tell it in the mediastinum between both the lungs. That portion, thymus, is going to be present. Thymus, what is going to happen? Only one thing is going to happen, that is going to be maturation of your T cells. Maturation of T cells. Now, students, thymus with age, it is going to degenerate. That means it is going to, the size of the thymus is going to reduce, and because of which at old age we do not have enough immunity like we have it now. So, it is going to reduce with age. Okay, reduce with age. That is, it is going to reduce, and because of which an old person will not have the same immunity that I have. Okay, yes, clear? 2 minutes breaker. I'll give you more than that. Okay, done, clear? This is clear? Okay, reduce with age. Now, second portion is your secondary lymphoid organ. So, let's assume this your bone marrow and thymus is the military camp. Let's assume that your thymus and bone marrow are the military camp. Okay, military camp. In the military camp, what happened? Training is going to happen. They are going to tell them how you have to encounter everything. Yes or no? Training is going to happen. So, this is your military camp. In the military camp, what has happened? Here, maturation and origin and maturation has happened. Okay, origin and maturation has happened. Now, from the military camp, we are going to send them to the battlefield, okay. From the military camp, we are going to send them, after we understood that, okay, proper training has been given, we are going to send them to the battlefield, and in the battlefield, what are they going to do? They are going to fight with the terrorist, right? They are going to fight with the terrorist. So, this battlefield is none other than your secondary lymphoid organ. That means everything, all the preparation happened in your bone marrow and thymus. Right now, we are going to send them, that is, the secondary lymphoid organ is the place where these people are going to actually face the antigen, okay, actually going to fight the antigen. Is this clear to all of you? Yes or no? So, secondary lymphoid organ, what is going to happen, students? They are actually going to encounter the pathogen, okay, encounter with the antigen. So, the battlefield where they are going to meet the terrorist, where they are going to fight the terrorist, is going to happen in your secondary lymphoid organ. In that, the most important secondary lymphoid organ is going to be spleen. Okay, why spleen is so important? Because it is the graveyard of your RBC. What is the lifespan of RBC? All of you in the chat box. Graveyard of RBC. We can also call it the blood bank. Okay, the blood bank. Students, tell me this. Graveyard of RBC. What is the lifespan of RBC in the chats? Quickly. Quick. How long the RBC's lifespan is there? Faster. Answers should come fast, okay? Fast, answers should come. All of you know this, so it is not that difficult. Just think about it. Once you think, you will know this. Yes. Very good, ma'am. Lifespan of RBC is going to be 120 days. Okay, lifespan is 120 days. Okay, perfect. Now student, the places where your mucus is there, okay, mucous membrane is there, there also we have our lymphocytes which is called malt. Okay, what is it? Malt. 50% of the lymphoid organs are covered by malt. You have to know the full form. Mucus, okay, associated lymphoid it covers how much? 50%. This number is important and this question has come many times. Okay, this question has come many times. What is the full form? Mucosa associated. Mucosa. Spelling is wrong. One second. Okay. Mucosa associated lymphoid tissue. Okay. Yes, all of you. Very good. Mucosa associated lymph tissue, lymphoid tissue. 50% of the lymphoid tissues are covered by your malt. This is also a protective covering in our body. Okay, that these places also lymph will be present. Done? Done. Thumbs up in the chat if you understood. Thumbs up in the chat if you understood. Quickly. Quick. Yes, done. Very nice, very nice. All of you, pakka. Okay, very nice. Thumbs. Okay. Okay, done. Okay. Now students, we have to learn more about your primary, okay, sorry, acquired immunity. Okay, two types of acquired immunity is going to be there. What are they? Two types of acquired immunity. Okay, we already learned acquired immunity is pathogen specific, very specific immunity. They are going to have memory. Now, under acquired immunity, two types of immunity that you are going to study. What are they? Humoral immunity, right? Humoral immunity. And one more, quickly in the chats, who is going to tell? Let me see who is that sharp student in the class. Humoral immunity and very good, cell mediated immunity. Okay, cell mediated immunity. Okay, now ma'am, what does this mean, ma'am? Is it something else that I am not able to, or will I not understand? Definitely you will understand. See students, humoral means fluid. Okay, fluid. So we can say that in the blood, okay, in the blood, okay, this is our blood vessels. In the fluid, we are going to have our B lymphocytes. What is going to be there? B lymphocytes are going to be there. Got it, all of you? Humoral immunity means what? Fluid, okay, blood. So, in the blood, you are going to see, you are going to find your B lymphocytes. And what are they going to do? I told you that B lymphocytes are going to form antibodies. So, they are going to protect our body, okay. So, humoral means again fluid, okay, fluid. And the fluid in our body is going to be blood and lymph, right? So, here we are talking about blood. So, what is going to happen in the blood? We are going to have your B cells, okay, B cells are going to be there and they are going to fight the pathogen by releasing antibodies. Now, what is cell mediated immunity then? Cell mediated immunity is where your T cells are going to be in place, right? T cells are going to be in place. So, here cell mediated immunity is mainly focused, we are talking about your T cells. Now, T cells directly does not form antibodies, but they are going to help, okay, they will help B cells form antibodies. They will help B cells form antibodies. Done. All of you clear? Yes. Okay. Now, we have some cells that you have to study, okay, but before that, I would like to ask all of you a Tattva question. Are you ready? All of you, Tattva question? Ready, steady, go. Okay, shall I ask you a question? or you're telling me, no ma'am, no need to ask us questions, okay? If you tell me that, I will not ask. No, I will still ask. Ready for the question? Okay, first question to all of you. Okay, this is questions that we haven't studied. Okay, tell me this. An example of innate immunity is, okay, option number A, PMNL neutrophils, B lymphocytes, T lymphocytes and T helper cells. Okay, T helper cells. So you tell me that an example of innate immunity is PMNL neutrophils, B lymphocytes, TH cells. Quickly tell me in the chats. Quickly tell me in the chats. What is the answer? A, B, C, D. Very easy question, no? Very, very easy question. Very good, very good, very good. I can see Ansari, Tanuja, Madhumathi, okay, Madre, Technoblade answering very nicely. Okay, very, very nicely I can see them answering. What does it mean, students? What is the question talking about? What is the question talking about? Yes, very nice. Okay, I'll keep asking you questions in the middle. Okay, now students, see, remember B cells, T cells are there. Okay, now we are going to just talk about their categories. Okay, they're going to talk about who and all comes under them, who are the people who are under them, right? So, okay, now here B cells are going to have two other categories. T cells will have more categories. Okay, now B cells, when I talk about, do you think they have memory? Do you think they have memory? All of you tell me in the chat box. Very good. PMNL, all of you understood, no? Neutrophils, polymorphonuclear leukocytes, or we can also tell cells. Okay, cells. Now tell me, all of you in the chat, quickly, what is B cells? Do they have memory? Yes, they're definitely going to have memory, right? So one cell of B cell is going to be the plasma cell. Okay, plasma cell, and one is going to be their memory cell. One is going to be their memory cell. Clear? So memory cell is going to do what? Obviously hold memory. Yes, they will remember that, okay, wait, I had done a primary response when this fellow came first time. Now I do not have to do a primary response because I have a memory of it. I will do a secondary response. Clear to all of you? Very, very good. Okay, again, this plasma cells, if you see in that way, they're not very specific. Okay, not so specific. Okay, now T cells. You think and tell me, what in all T cells have you heard about? Now T cell, students, we have T helper cells, T helper cells, T suppressor cells. I don't think I'll be able to fit everything here. Okay. One more I have to write. So write it down right now. I will, move it down otherwise. I have to write it again, otherwise there's no place. Okay. T killer cells are there, T helper cells are there, T suppressor cells are there, and the last one is T memory cells. Okay. So if all of you can write that, or let's try to fill it here only, T memory cells. Okay. T memory cells. So what and all we have, students, under T cells? We have T memory cells, we have T helper cells, T suppressor cells, and T killer cells. Okay. Now T killer cells, we will use it for graft rejection. All done? Clear? Very good. TK killer cells. Yes. Killer cells, memory cell, helper cell, and suppressor cell. B cells, we have plasma cell and memory cell. Now, important part of this is learning about antibodies, right? Antibodies. Now students, remember, your T helper cells will have your CD4 receptor, which you're going to study later in your HIV. So I'm going to just write it here only. Let's say this is our T helper cells. Okay, this is our T helper cells. I'm going to just draw it here. Okay, so it will be easy for me when I'm teaching you HIV as well. Okay, this is our T helper cells. Okay, let's shade it. Oh. Okay, this is our T helper cells. I told you what? They have CD4 receptors. Okay, they have CD4 receptors. Now, what is CD4 receptor? Clusters of differentiation, we tell the name for it. So, here this is CD4 receptors, and the CD4 receptors are not very simple fellow. Okay, they are not very simple fellow. Why they are not very simple fellow? Because CD4 receptor place only your HIV virus is going to come and bind. Be it your dendritic cell, be it your T helper cells, macrophages, CD4 receptors are the place where your HIV will come and bind, and that is present in your T helper cells. Okay, full form of it is cluster of differentiation. Cluster of differentiation. Done. Is this clear? So, when I talk about CD4 receptor, don't stare at me that time. Okay, HIV, when I am teaching you HIV, don't look at me like full in shock. Okay, this is why I am introducing you here only. CD4 receptors are present on the T helper cells where your HIV will come and bind. Done? Yes, very disappointing seeing all the conjugates. My students are only getting disappointed. So, imagine my situation. Okay, imagine my situation. Right. Okay, shall we study structure of antibody? Okay, structure of antibody, or do you people want a 2 minutes break? Okay, do you want a 2 minutes break? We can take a 2 minutes break and come back and start with antibodies. Okay, yes, quickly tell me in the chats. Or all of you drink water and take a break. For few days I will not strain my throat, students. Okay, so then without break we can teach for longer time, otherwise that's all gone. One shot throat will go. Okay, yes, water break. Okay, 2 minutes. Okay. Got it? Just 2 minutes break, just drink water. Don't go anywhere, just stay here only. Go drink water and come. Let the live keep running. Where you people are going? Okay, let's continue. Okay, voice is clear now, students. Okay, now let's do antibody. Okay, let's do antibody. So far everything is clear, right? No doubts so far? Pakka no doubts? Later don't tell me, ma'am, doubt was there, ma'am. Okay, tell me right now if you have any doubt. Okay, now students, when we go talk about antibody, first we have to remember what is antibody? What is antibody? Someone who is going to fight the antigens, right? Someone who is going to fight the antigens. Now, what is antigens? Any foreign particle we can call an antigen, right? A foreign particle is called as an antigen, a disease causing one. Now, someone who is going to fight the antigen is called as your antibody. Now, antibody, when we talk about, you should understand from where did we derive this, okay, or who is forming this. Now, remember all of you, yes or no, all of you remember this one thing when we learnt about plasma, when we studied about body fluids and circulation, we had spoke about globulin proteins. Yes or no? How many of you remember this? Globulin proteins. Raise your hands those who remember this word, globulin proteins. And we spoke about immunoglobulins, right? So globuloproteins, especially the one that is going to form is going to be your gamma, okay, gamma globulins. Gamma globulins. Now students, remember that this globulins, where we spoke that they are going to form immunoglobulins, right? What is the antibody? We speak about IgA, IgG, IgM, IgD, immunoglobulin A, immunoglobulin G, right? So these globular proteins were the ones, right, who are going to do this purpose, do the formation. Yes, globulins, okay? You go to globin, for you people know, only storytelling is important. Yes, for you storytelling is the most important thing ever. Storytelling. Yes, this is clear. Now, students, remember globulin proteins, gamma globulins. Now, these people are going to form your antibodies, and these antibodies, okay, these antibodies are formed by whom? Formed by whom? B cells, plasma cells of the B cells, okay. Plasma cells of the B cells is going to form these antibodies, and who is going to help? T cells is going to help, but T cells are not going to form anything, okay. GM express, the whole thing is GM express only. From that time I am only writing no, right? Yes or no? I am only writing full thing is GM express, correct? Yes, Selvi is asking where is GM express? Full thing is GM express, dear. Yes or no? Yes, very good. Okay, done. Now students, remember, okay, remember that antibodies are formed by your plasma cells of the B cells. Now these antibodies are going to be formed at a very faster rate. Our body is going to form it. Now, how many chains do they have? All that you have to know, no? So first we have to draw the antibody, okay. Shall I draw it in this side only? Okay, I think I will just erase this. Okay, I will just erase this and draw it here only. Okay, so we can write everything in one sheet. So I am going to write here whatever I wrote, okay. Antibodies are formed by your gamma proteins. Okay, that I had already written, no? Antibodies okay, done. Now we have to draw the antigen, okay. Draw the antigen, right. So let's draw the antigen now, okay. All of you draw it with me, okay, draw it with me. Okay, does it look neat? Okay, done. Now students, this is the fastest I have drawn an antibody, sorry. Now, 1 second, just give me 1 minute. I need some more space here. Okay, yeah. Okay, these are your disulfide bonds. Okay, that's why I took some space. I don't think it is seen. S you can see here. S you can see. Okay, now students, let's draw a few more things here. Okay, let's draw a few more things here. Yes, can you see now? Yes, this is done. Okay, this is the fastest antibody. This is just to make you understand. Now students, if you look at this antibody, you are going to see 4 chains, right? But see, this is actually 2 heavy chains and 2 light chains. So we can write the formula for antibody as H2L2. Can I write it H2L2? Yes or no? H2L2, what does it stand for? 2 heavy chains, 2 heavy chains and 2 light chains. Now this heavy chains, light chains, all of them are clubbed together, kept together with the help of disulfide bond. Kept together with what? Disulfide bond. Can you see one disulfide bond I have written? They are clubbed by disulfide bond, that is SS bond. SS bond. This is called the disulfide bond. Done, done, done. All of you, quickly tell me in the chat box if we are understanding everything. Disulfide bond is clear, light chain is clear, heavy chain is clear. Now which are the heavy chains? These are the heavy chains. These 2 are the light chains. So shall I label them? This is your light chain. Okay, light chain. And this is your heavy chain. Heavy chain. Clear? Now I am not going to draw all the disulfide bonds, okay, because it will take a lot of time. So I am not going to draw that. Is that okay with all of you? Yes, okay, right? Fine. Now students, remember that in your antibodies there are hyper variable regions, variable regions. So these portion here are going to be called your variable regions, which usually will be drawn in a different color. Okay, since I am not going to draw that, I will just write that this regions, okay, this portion, no, this will go wrong. Okay, I will just label like this. These are your hyper, okay, this whole region, this two portions, okay, hyper variable regions. Okay, and these regions, right, these regions are going to be your variable regions, and this region is going to be a constant region. Okay, variable region. Clear to all of you? This down portion is going to be a constant region. This too is going to be a hyper variable region. This is going to be a variable region. Now, very, very important thing. Let's draw an antigen first. Okay, let's draw an antigen, all of you with me. Okay, this is your antigen. This is your antigen. You might tell me, ma'am, it looks like a cap. You're not supposed to tell that, okay? Don't be disrespectful for teachers' diagrams. Okay, so this is your antigen. Okay, yes, this is your antigen. Okay, which looks beautiful. It looks like a mushroom. Oh, target completed. Very nice, very nice. So this portion of the antigen. Okay, I am telling you this is an antigen. Shall I write everything about antigen in a different color so you will be able to differentiate? This is an antigen. On the antigen, there is a region that can go and bind with the antibody, which is called the epitope. Which is called the epitope. Clear to all of you? The region on the antigen which can go and bind with the antibody is called the epitope. So there should be a region in the antibody where the antigen can come and bind. Exactly that region. This region is called as a paratope. This region is called as a paratope. Oh no, something I did. Okay, this region is called the paratope. In bracket you can write antigen binding site. Okay, Many students get paratope and epitope confusing. Please understand the region on the antibody where the antigen can come and bind, that is called the paratope. On the antigen, what do they have? They have the epitope. Clear? Paratope present on the antibody, epitope present on the antigen. So paratope, epitope will bind, and that is how antibody is going to attack the antigen. Is this clear to all of you? So we will have two paratopes in one antibody. We will have two paratopes. This is paratope 1, this is paratope 2. So two paratopes in one antibody. Clear? So far good? Everything is clear. H2L2 is clear. Very, very important. Two heavy chains, two light chains. They are bound by disulfide bond. How many disulfide bond? 16 disulfide bond. All of you can count in your NCERT if we have 16 or not. Okay. You can count in your NCERT if we have 16 disulfide bonds or not. Count and tell me if I am correct. Or do we have 14? Okay. Count and tell. Take your time. Count and tell me. Quickly. Okay, and students, one more thing, I saw one question. I think biomolecules, you might learn it when we talk about amino acids. No, see, these are proteins, right? These are proteins, so they are made up of what? Amino acid chains. So amino acid chains will always have a C and an N terminal, right? N C terminal. Yes or no? So in this, they will ask you in an antibody which is the C terminal, which is the N terminal. So you have to remember that this is the N terminal, okay, this one, and this is the C terminal. Clear? Again, same thing goes here, N terminal, C terminal, okay. N terminal, C terminal. So that is the N terminal and this is the C terminal. Clear? Yes or no? So many things to remember. It's easy only, no? N terminal and C terminal. Anyone has any doubts? Tell me. If you have doubts, I am ready to explain it once again. All I want you to do is understand it first. Okay, understand properly so it is easy. Okay. Now students, we can tell that your antibody, okay, these are additional some terminologies, just remember it, okay, that this entire thing, how does it look like? It looks like a Y-shaped structure. Do you want me to write everything about antibody in the next sheet? I can do that, okay. I wish I could copy paste this, but I cannot copy paste. That is a problem. Shall I do one thing? Shall I write about antibody in a previous page and I will just continue in this page only, okay. I'm just going to erase this. Okay, and we'll write it in a small box later. Now, what did I tell? I told you that this, if you look at the structure, what do you understand from looking at the structure? You understand that the structure is a Y-shaped structure, right? So, when we talk about antibody, okay, when we talk about antibody, we talk that this way, the question can come, it is a Y-shaped, okay, it is a Y-shaped tetrapeptide chain. Why am I telling it's a tetrapeptide chain? Can you all tell me? Because there are 4 chains, right? 2 light chains and 2 heavy chains. So it is a tetrapeptide molecule. Tetrapeptide molecule. Why did I say tetrapeptide? Because of the fact that they have 2 heavy chains, okay, 2 heavy chains and 2 light chains. Okay, 2 light chains. And remember students, they are made up of amino acids, so it is going to have N terminal. Okay, you would have studied this in biomolecules, amino acid structure, and they are going to end in your C terminal. Okay, so N terminal and C terminal. This question I have seen some places, that's why, yes, 220 amino acids and all, we talk about 440, 220, but right now we are not going to tell that. Okay. Other things are clear, and they are bound by disulfide bond. How many disulfide bonds? Okay, each chain is bound by disulfide bonds. Did you all count? Did you all count? Is it 16? They are bound by disulfide bond, that is SS bonds. Okay, SS bonds, and it is around 16 of them. Okay, 16 bonds are present. Clear? Perfectly clear, everyone, or only some people understood? No doubt. Okay, no doubt. Yes, perfect. Yes. Now students, since they have two paratopes, okay, paratope, all of you remember, no? Since they have two paratope, we use the word bivalent. This also I have seen in some places, okay. Since they have a presence of two paratope, that means two antigen binding sites, okay, I am writing it in bracket, two antigen binding site, okay, we can also call them bivalent. Bivalent. Done, 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 done. Okay, no doubts, right? Perfect. Antigen, antibody is clear. Now we will just write whatever we erased, okay, just in one corner I will just write that also, then we are done with antibody. Okay, so not done like this portion is done. Okay, they will fight. Students, all these notes will be available on the Telegram channel. You can see the Telegram channel name there, as well as Vedantu NEET English Telegram channel. Notes will be available. Tejas also, yesterday's Tejas series also, I will be sending it to all of you, okay. Fight against antigen. Okay, and they are formed from your globulin proteins. Exactly which globulin protein, if they ask you, you are going to write gamma proteins, okay. These are small additional information. It's not like, my God, ma'am, so much you taught out of NCERT. Okay. Gamma globulins. And who are going to release the antibodies? Antibodies are going to be released by your B cells. Okay. B cells. Which B cells? Plasma cells of the B cells are going to release your antibodies. Okay. Release the antibodies. Clear? Clear? Yes. Okay, I'll give you a break. Lunch break. Any doubts? Shall I ask you a question? Let me check your Tattva. Yes, antibody questions are there. See students, they can directly give you the diagram and ask you to, label the diagram, okay. Other than that, they can ask you like this. See, the 4 polypeptide chains of the antibodies are made up of option A, okay. So, the 4 polypeptide chains of the antibodies are made up of option A, 3 heavy chains, 1 light chain, 2 heavy chains, 2 light chains, 1 heavy chain, 3 light chain, none of the above. A, B, C, D. Okay, A for 3 heavy chain, 1 light chain, B is 2 heavy chain, 2 light chain, C is 1 heavy chain, 3 light chain, D is none of the above. So, tell me the answer for that. Tell me the answer for that. After that, I will ask you one more question. But I have to teach you little more, but yeah, I will ask you one more question. Okay, this is from Tattva. Okay, this is your Tattva book and you can see that human health and diseases backside questions are there. Okay, I am asking you from this questions. Yes, what's the answer? Most of you are telling option number B. Very good. 2 heavy chain, 2 light chain. Next question. An antiviral substance produced in response to the viral infection for restriction. Okay, its multiplication is, okay, it's an exemplar question. An antiviral substance produced in response to viral infection. Option A, antigen. Option B, antiviron. Option C, interferon. Option D, viron. Okay. Yes. Antigen, antiviron, interferon and viroids. Option, the question is an antiviral substance which is produced. Tell me quickly. Quickly the answer should come. Okay. Yes. Quick. Tejas book is there for Tejas series. No, now we are doing RNA series. So, we are going to use Tattva. Got it? I am asking you questions from Tattva. So, tell me the answers. What is the answer? Option A, option B, option C, option D. Quickly answer. I am checking for more questions. Yes, very good, very good. I can see few people have grasped the question well. The answer is going to be interferons, right? The answer is going to be interferons. Clear? Yes? Interferons. Very good, okay? Because the antiviral substance that is released is going to be interferon. So, students, no doubts, no? Up to this, see so much we have written. I hope till here no doubts are there. Okay, so now we have to learn different types of antibody. Okay, different types of antibody. which I've taught you in many other classes also. So I'm just going to give an overview. Okay, I'm going to give an overview. Before that, we can have a lunch break and come back, or after this we can directly finish the class. Okay, so you tell me. Yes, tell me quickly. I'm waiting for your answers. Answers. What is your answer? Fine, we'll continue. Okay, now remember that antibodies, okay, when we talk about antibodies, students, there are 4 types of antibodies that you have to remember, and you're going to remember the word gamed, okay, gamed, that is G-A-M-E is gamed. Okay, add one D to it and we make a non-existing word from Oxford Dictionary. So gamed stands for IgG, okay, IgE, IgM, IgE and IgD. Clear? IgD, IgE, IgM, IgG and IgE. These are your 5 types of antibodies. Clear? Now, in this, the most common question that I have seen is about your IgG, the one who is going to cross the placenta. That means from the mother through the placenta, the antibody that is going to go to the baby, which is that IgG. Okay? All of you remember this? Yes or no? Last time also I had made you write the whole paragraph. Correct? Very good, very good. Most of you are able to understand that. Now students, also one more question that comes majorly. I am not going to go into the details of this because again I am not doing the whole chapter. Most common questions, only that we are going to see. Okay. So the most common question that I have seen is which is higher in number, the percentage wise, who is the most, okay, most common one. So that questions I am going to write here, and it is also there in Tattva as well. So the most high, okay, and that means the number wise, who is going to be high? All of you go and open your notes and see what is the most one in number. Okay, next question I have seen, the one who can cross the placenta. Okay, cross placenta. Next question I have seen, the largest one. Okay, in the terms of size, largest in size. These are the most common questions that I have seen. So you have to answer me one question, okay, quickly, and one more question which is actually connected again in body fluids and circulation also study, which is the one that is going to be in the colostrum, or colostrum, which is going to be the antibodies. Okay, this is the most common questions. Now if I open Tattva and check also, these are the questions that are going to be there. See, I will pick one question randomly and you will see, which of the following immunoglobulin does constitute the largest percentage in human milk? And this question has come in NEET 2015. Okay, NEET 2015. Got it? The question is, which of the following immunoglobulin does constitute the largest percentage of human milk? First question. Okay, already so fast I spotted the question. Now if I go further and check, there will be a lot of questions, same types of question only will come. So just here and there pattern will be changed a little. Okay, pattern, that side, this side will be changed, otherwise the questions are going to be same itself. See, let's see. Okay, there's one more question. The antibody which can cross placental barrier. Okay, antibody which can cross placental barrier. This is also a question. Okay, this is also a question. So can we answer one by one? Can we answer one by one, all of you? Yes? Quickly tell me which is the highest in percentage. After that we will be able to finish the chapter fast and furious. Yes or no? Fast and furious. Okay, so we will start from the first question. So students, most high in number is going to be none other than your IgG. Okay, what is this? This immunoglobulins. Okay, immunoglobulins, because again it is from the globulin protein. So the word immunoglobulin, again they are going to help in the immunity. So remember like this, answer is going to be IgG. Which can cross the placenta? Again your IgG can cross the placenta. Now largest in size is going to be your IgM, right? IgM, because again it is made up of 5 Y-shaped structures. All of you, very nice, very nice. I can't write IgA. Where? Here I can write IgA. Okay, here I can write IgA. Did I forget to write A? Oh, that's what you're telling. Okay, A. IgA. Okay, yeah. So here colostrum, the one in colostrum is going to be IgA. These are your most common questions. So learn these questions. Okay, learn these questions. Is this clear? Okay, yes, I have written, students. I have written. Okay, don't worry. Don't worry. I have written it right now. Just made a correction. Okay, this is a pentamer. Okay, pentamer. So that means what? Like this. Okay. Okay. Like this. Like this. Like this and like this. Okay, like this. So the pentamer structure is there. Obviously the structure is not perfect. So pentamer, it is made up of pentamer. Again, J-shaped chain will be there, which I have taught you in previously. So you can check out that lecture as well. Okay, J chains will be there to keep them together. J chains. So common questions are done. That's all I'm going to teach. I'm not going to teach in depth. Okay. Now if you want to know the percentage, this is going to be, you can write at the range of 75% to 80%. Okay, 80%. This is the range of it. That is why they are the largest in number. Cross, who is going to cross the placental barrier? Your IgG. Largest in size, IgM, because they are made up of pentamers. Colostrum, who is going to pass? IgA. Done, done, done. All of you, any more questions should I search? I think this is fine, right? This is fine, no? We have searched the questions and we have found the answers as well. Perfect. Okay, done. Okay, done. Clear? Done, done, done. So questions that can come from your antibodies I have already discussed. See, all this entire slide, any question can come. Any question can come, including your N terminus and C terminus. Any question can come. It can be asked about your disulfide bond. It can be asked about your hypervariable, variable regions. Again, FAB regions and FC regions also are there. But it's fine for you to not know. But in case someone asks you suddenly, like, what is it? Understand, these are your FAB region, this is your FC region. Again, the constant regions, okay? They are the constant region. Full form is little more different, but otherwise, this is the FAB region, this is the FC region, the constant region, this is the variable regions, okay? Now, heavy chain, light chain, epitope, paratope, they can ask you. One antibody has how many paratopes, they can ask you. In IG, they can also ask you one different question in IgM. Okay, how many paratopes are there? So all that you have to count and tell, okay, because if one antibody has 2 paratopes, then 5 antibodies together will have 10 paratopes. Like that questions also can come. Okay, but these are the most common questions. So I will just write it here. Okay, most common NEET questions. Okay, NEET questions, most common ones. So how are you going to remember it? Gamed. Gamed you are going to remember. Okay, gamed. Done? Done, done, done. Okay, now we will resume after a break. I am thinking that actually, I think I will teach one more topic. I'll teach you active and passive immunity. So today we will finish immunity portion, okay, and vaccination, immunization also I'll just tell you. After that, I will do AIDS, cancer and the diseases together in one part. Does that work for all of you? Okay, I will do that also together. Diseases, see, I am not going to tell you all the common diseases, typhoid, this, only malaria we have to concentrate more, okay. Then we will do AIDS, cancer, clear. So take a cutie break and come back, okay, take a lunch break because all of you are crying that you are hungry. I'll also eat something and come. What's the time now? Okay, let's assume 2 o'clock itself. So come back by 15 minutes is enough. Okay, 15 minutes. I want all of you to come back, okay. Don't just 5 people come back and then the rest of them go. All of you should come back because again we have active and passive immunity. Lot of questions come from this also. I'll ask you Tattva questions as well. Clear? Fine. This gives a feeling of a, you know, offline class. No, the teacher will have the book in the hand. They will ask you questions. This is important, okay. Yes, so today we will complete immunity, totally will complete immunity up to active and passive immunity, and then we will do a quick recap, okay, from the starting till the end, and then we will do AIDS, cancer and malaria together, okay, because I am not going to go into the common diseases anyways. If you want, I can just write a tabular column and you can just see the tabular column, okay, otherwise we are going to do this. Clear? Yes? Yes. Okay, Newton's, this Newton's head, instead of apple, some other thing should have fallen, okay, coconut or, you know, jackfruit or something. That's why He's telling if you want apple, just take it from my name. Wow, wow. Okay, this is why students pray when they are learning that in Newton said something else should have fallen. It's because of people like you people, okay, who spoil Newton's name. So come back, okay? Pakka should come back by 2:15. Yes, I don't know why. I thought it was 220. And then I'm like, wait, did I tell 215? Okay. So by the time I went out only it became 225. Okay, so sorry. I forgot the time what I wrote. Okay, shall we continue? Yes? Okay, chalo. Thought it was 220. Okay, yes. Target done. Huh? Yes, this is also done. Okay. So, students, all these names are there. Okay. IgG stands for immunoglobulin gamma and all, but it is fine. Okay, if you do not remember it, that also. Now, one more topic we have is tissue transplantation. Okay, that also just 1-2 minutes it will take. We'll finish that. After that, we'll go to active and passive immunity. Got it? So, we'll divide the page into two. Yes. So, students, tissue, that is your graft rejection and all we have spoke, no, transplantation and all of that. Okay, that is what we are talking about. So, I'm going to write tissue graft. Do not be like, what is this new word, ma'am? Same thing. Okay, graft and transplantation. Okay, this is the topic that we are left with. Now, students, whenever tissue, okay, let's say when we need an organ, okay, one kidney failed, both the kidneys failed, we have to take a transplantation. What are the things that you need to check on? First thing you need to check on is that if the blood group is matching, right? Blood group is matching. So, the first thing that you are going to do ever before this graft implantation is going to be blood group matching. Second thing that we are going to do is called as MHC technique, which is actually used to reduce, okay, maximum, it's actually a shortcut to rejection. Okay, so the body shouldn't reject, no. So for that we do a major histocompatibility test, that is your MHC. Now MHC actually reduces the chance of graft getting rejected, which is actually, you know, if you read research papers, you will understand that it is told as a shortcut, okay, to avoid rejection. Okay, so just remember the word MHC. Can you all remember the word MHC? Yes or no? What is the full form? Major histocompatibility complex. Okay, major histocompatibility complex. Actually this you will study in detail obviously in your MBBS, but major, okay, major histocompatibility complex. Okay, can you remember MHC? Just remember the short form. You don't even have to remember the full form, just remember the short form. Can you do that? It's also called as HLA. Okay, don't write HAL, HLA. Okay, human leukocyte antigen. Okay, again short form I am writing here. If you can remember the short form, it will be very, very good. So students, after this, the last thing that you do is you use an immunosuppressive agent, right? You use an immunosuppressant or an immunosuppressive agent. What is the agent name? All of you tell me in the chats. We have learned this in microbes chapter. Immunosuppressant that will make sure that the graft rejection does not happen, right? The graft rejection shouldn't happen, so that we are going to use an immunosuppressive agent or an immunosuppressant. What is the name of the immunosuppressant? All of you in the chats. Quickly answer. I have taught this in microbes, so I am not going to explain. Okay, I am not going to explain. Tell me in microbes chapter we have learned. All lazy people, can't remember one thing. Yes, tell fast, quickly. Very good, very good. I can see one answer, that is Madhumati has answered cyclosporin A. Okay, cyclosporin A. Clear? Students, there is an antibody mediated rejection also, which is called as AMR. So if you see anywhere in the coaching module, anywhere when you are reading, if you see the word AMR, understand antibody mediated rejection. That rejection also can happen, okay, in our body. When we are taking a graft from another person, when we are taking an organ from another person, it is usual that the body is going to reject it because it's a foreign particle. To reduce the rejection, we are going to use MHC, that is major histocompatibility test, or HLA. play, okay. And at the same time we are also going to use immunosuppressive agent because of the use of immunosuppressive agent nowadays, which has become very common, and because the technology also has increased, graft rejection also has reduced. But it was not the same scenario back then when immunosuppressant wasn't found. Clear? Okay. Now next topic is going to be active and passive immunity, okay. Now what is active immunity, what is passive immunity? Whenever we talk about active and passive, active means what? We always tell that use of energy is there, ATP is being used, okay. Passive is what? That means ready-made, okay, ready-made. What happened, Tanuja? Ready-made. So as active and passive here, when we talk about, it's the same thing, okay. So first thing is called as active immunity. That means our body itself have to make this, okay. And second one is called passive immunity. Passive immunity. So active immunity means what, students? The body has to make it. It has to take something. It has to take the energy and make it out, right? It has to make it. So here active immunity, passive immunity. Passive means a ready-made form of immunity. That means I made it in some other animal and I am just injecting it into my body. The antibodies are already made, so it is why it is called passive immunity. That means ready-made antibodies are coming. Okay, ready-made antibodies are coming. So this I can call it as a ready-made process. Okay, ready-made process. Clear to all of you? Yes or no? Easy peasy lemon squeezy? In the chats please answer. Is it clear or not? Then only you will understand this. Lot of questions come from these topics. So this topic is very important. Okay, very important. Yes, tell me primary, okay. Okay, done. Now, students, active immunity can be further divided, okay, active immunity can be further divided, that is we have to form, okay, formed in the body, okay, formed in the body. Now, passive immunity is ready-made immunity, or we can also call it as preformed, okay, preformed antibodies. I am using NCERT words only, preformed antibodies. Now, these topics can be further divided into active and passive immunity can be further divided into what? Happy Diwali to you also, Chaitanya. Okay, active immunity can be further divided into natural and artificial. So, question is going to come from here. Okay, artificial. You have to know what is this under each of them. Okay, again this also I am dividing it into natural and artificial. Can you think and tell me what it could be? Can you think and tell me what it could be? Say students, natural immunity means what? Now I got exposed to a pathogen. Okay, let's say this is, okay, this is Mintu, this is Mintu. This is Mintu and Mintu got exposed to a pathogen. This is the pathogen. Now, the moment Mintu gets exposed to the pathogen, the body will automatically start forming antibodies because we have immunity, yes or no. And students, by the way, this is a cell mediated immunity, okay, CMI. This is a cell mediated immunity, just suddenly I remembered, okay. Now, students, when this person, the Mintu, is going to be exposed with a pathogen, the body is going to automatically make antibodies, which is coming under natural, yes or no. All of you tell me in the chat box. Mintu got exposed to a pathogen and automatically body will start forming the primary and secondary response. So, primary response happens, automatically antibodies are going to be formed. So, this is going to fall under your active immunity natural example, okay. Now, artificial means what, students? Let's say I give a vaccination or I get a vaccination. What is a vaccination? It could be a vaccination, could be a live attenuated one or a let's say, we cannot say completely like inactive one, okay. Inactive form of a pathogen will be in my vaccination. The moment it enters into my body, the body is going to start forming antibodies. Isn't it an artificial way? Because I am injecting an inactive form into my body. I am injecting an inactive antigen into my body so my body can make antibodies. Is this clear to all of you? Please listen to me. Diwali will come and go. Your NEET will come only once. Now write this. Tell me this. Is this clear? Fast. Okay, so in vaccination what is happening? Weakened antigen. Okay, weakened antigen is used. Okay, the inactive form or the weakened one. The moment this enters into my body is going to, okay, the moment this one enters, okay, into my body, what is going to happen? My body is going to start forming antibodies. My body is going to start forming antibodies. Clear? This is clear? Yes? Okay, done. Now, under natural passive, passive is already understood that it is ready made. Now, let us say the natural is going to be, students, colostrum. Okay, from the colostrum, the mother is going to give IgA to the baby, right? IgA antibody is given to the baby. Now, this is a natural form of passive immunity. That means we have not injected anything into the mother. The mother has naturally formed the colostrum, right? Without an injection from anything outside, the mother has naturally formed it. But the baby is getting it as ready-made. So, I will put it under passive natural immunity. Now, passive artificial, students, let's say snake venom. Okay. When we take snake, when we get bitten by a snake, snake venom has entered the body. So, for that we will have to give what? Anti-venom. This anti-venom is actually made in horse, okay. That is, the venom will be injected into horse body and the horse will make ready-made antibodies. That antibodies will be injected to a person who has got bit by a snake. Understood? So, this is a complete passive method, complete artificial method. Antibody is made in someone else's body, it is only injected into my body. Is this clear to all of you? Questions will come from immunity, active and passive immunity. I am telling again and again, so please be careful. Okay, please be careful. Let me see questions on that as well. It's a very small topic, okay. See students, one question has come, it's a big question. Which of the following is true regarding organ transplantation is the question, okay. Histocompatibility antigen. See, this is why I mentioned this HMC here, because I have seen in most of the coaching module they have used this word, but in NCERT also it is used. It's not like it's not used, but it is little in a loophole, so you will not understand. See, histocompatibility antigen and immunosuppressants are used to suppress immune response, passive immune response. I'll send the pic of this in the Telegram, okay. This picture will be sent on Telegram, so you can solve it on the Telegram channel. Clear to all of you? Lot of questions have come from there. Then students, I think immunization question has come. Immunization is achieved by successful delivery of vaccines. Vaccine is a preparation. Passive immunity is defined as what immunity? Okay. And when a host is exposed to antigen which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body. This type of immunity is called active immunity, passive immunity, autoimmunity, allergy. Can you answer this? All of you tell me. Tell me. Tell me the answer for the students. When a host is exposed to antigen which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body. This type of immunity is called as active immunity, passive immunity, autoimmunity and allergy immunity. Okay. So, does it fall under active immunity or passive immunity? Tell the answer and tell me. Just check the answer, all of you in the chat box. Quickly. Active immunity. Yes, they are active immunity. Why? Because we have already mentioned that vaccination is a weakened antigen. It could be a protein, it could be a dead microbe, it could be a live attenuated one. But when it enters our body, antibody is not ready made. Antigen is being exposed. Okay, automatically body will start forming antibody. So it is going to be active immunity. Clear to all of you? Yes or no? So such kind of questions can come. So you should be aware of what your answer is going to be. Is this clear? Okay, and last topic for the day, students, it is going to be allergy. Okay, last topic for the day is going to be allergy. Immunity, everything is clear to all of you. Anyone has any doubts, please ask me right now before I move to the next topic. Okay, vaccination and immunization is a small topic, you can all learn it. Okay, now students, allergy means what? Any disturbance, right? Any disturbance in our body due to a allergen. Allergen means what? Someone that can cause allergy. Okay, typhoid, active, vaccine, active. Okay, so allergy here students, what does it mean? It is caused because of an allergen. It is caused by an allergen. Yes or no? Allergen is what? A potential person, a potential thing. Okay, it could be pollen grains, it could be hay, it could be any of it which can cause me an allergy. Okay, that is an allergen. Now, when you talk about allergy, do you remember the cells that are going to respond? All of you in the chat box, tell me which are the cells that are going to respond. So, students, in the first exposure, okay, in the first exposure, what is going to happen? Your B cells, exactly your plasma cells of the B cells, obviously what is going to happen? They are going to release your mast cells. Okay, tummies, I have already told the answer here. Okay, now students remember this B cells, plasma cells, going to release your IgE, immunoglobulin E, and again main character is your mast cells. Okay, mast cells. This is the most important heroine of the conversation, your mast cells. These mast cells are going to help us understand that they are going to cause the inflammation. Vasodilation will happen. That vasodilation will let more blood to enter, and because of which we will start seeing redness. Okay, we will start seeing redness. Clear? Yes. Clear to all of you? Yes or no? What are you people telling? Tamil language you are telling? Yes. So, this mast cells will get activated. Okay, they will get activated and they will, I am waiting for your response, that only half of the time will go. People are responding so slow. Okay, they are going to release your histamine. Okay, serotonin. Right. Now, what are these people going to do? They are going to do vasodilation and vasoconstriction. Yes or no? Agreed all of you? Yes. So, what are they going to do students? They are going to do vasodilation. As they do vasodilation, automatically, okay, automatically blood will start flowing. So, let us say this is our blood vessel initially. Okay, this is our blood vessel initially. Okay, what they are going to do? They are going to do vasodilation. That is, they are going to make the blood vessels here, okay, increase in size so that more blood can enter. When more blood enters, what will happen? Automatically you will start seeing inflammation and redness. Inflammation and redness, making you realize that you have an allergy with the thing that you have consumed. It could be pollen, it could be hay, it could be even, you know, some, this nuts. Okay, it could be pollen, it could be hay, it could be nuts, it could be milk products. I am writing the common ones, okay, that causes allergy. It could be milk products, it could be some, you know, vegetables and fruits. It's very less, but mostly, okay, you will have with these things, okay, these things, okay, lactose intolerant and all you have heard. No, this lactose intolerance is how? Because of milk product, but that doesn't have anything to do with. Some people it will turn out to be very massive allergy, like I have seen most of the people when they eat peanuts or walnuts they get allergy. That means the entire body will have rashes, okay. But lactose intolerance, where you go to a uncomfortable stage, where you feel abdominal pain, you feel like you have to puke, you feel tired, you start sweating, that is different, okay. Clear? Live is slower, that is why I am not able to see the chats faster. Okay, that is why I am not able to see the chats faster. Yes or no? Okay, yes, it is a hypersensitive immune response. That is why it is very important for you to remember. Okay, it is a hypersensitive immune response. Okay, very nice. Okay, so I will write it here. hypersensitive immune response. So students, if you get an allergy, what can you take? You can take the drugs like, drugs means medicines, okay? Don't get excited. Drugs like your antihistamine, some steroids, okay, adrenaline based ones. These will reduce the histamine, okay, it will reduce the inflammation and redness. Clear to all of you? It will reduce the inflammation and redness. These are your drugs also, okay, stored drugs. Done. Clear? Allergy is clear. Mast cells, don't forget, this is the most important cell. Mast cells like this will be there, it will get activated and they will release your histamine. Histamine is the one which will do vasodilation, and because of that, okay, because of that, they are going to cause redness and, okay, redness and inflammation. Done, done, all of you? Done, done. Clear? Allergy is clear. Vaccination and active and passive is clear. All of you? Let's take an overview of what and all we learned. Ig, immunoglobulins are clear. Antibody is clear. Okay, then we studied acquired immunity. We studied B cells and T cells. Then we have studied primary and secondary lymphoid organs. We studied acquired immunity. We studied about cytokine barriers, physiological barriers, your cellular barriers, okay, cellular barriers. We studied what is barriers, what is physical barriers. We studied first line of defense, second line of defense, and we studied what is health. Clear to students? All of you, is it clear? Everything, all the topics are clear to all of you? Is there anyone who doesn't understand anything? I'll share this notes on Telegram, okay. Before that, I want all of you to know that the Diwali combo offers are still running. So, I want all of you to make your decision wisely and take this at 2500. Now, I tell you what is the use or how you can go forward. See students, this is a batch, okay. You will get all the recorded lectures, okay, because everything happened live, but since you are joining now, you will get the recorded lectures. Now, even though you are not back in your syllabus because you do not have backlogs because you are attending our YouTube live. Now, what you can do is you can go and watch the recorded lecture whenever you have a doubt. All the lectures are in a span of 1.5 hours, okay, and you will also get free notes, very good notes you are going to get. You will get a lot of e-books where you can practice a lot of questions. You will get Tattva. So, if you purchase all of this individually, then you can never get it in this price. Now, what is NYTS? You will get 25 total tests, okay, 7 full syllabus and 12 part syllabus and 6 subject wise tests. And you will also get detailed video solutions, you will also get exam experience, as well as you will get your 360 performance, how you are performing compared to the other students in our batches. Okay, so take this at 2500 is right now the price. Definitely after Diwali the prices are going to change. Now you are telling me, ma'am, I don't want NYTS, then you can go for only DNA batch where you will get recorded lectures, where you can also get mentorship and guidance. We take mentorship and guidance classes, plus you will also get all the Tattva e-books. For the books at least you know take the batch. You will get all the e-books because we have taken the partnership with most of the books. So, we have the copies of the e-books, okay, and almost all the e-books are your question based e-books. So, you can practice a lot of questions on a daily basis for this price. Link is in the description. I will show you the link as well so that you can go and register. Now, students, this is the video all of you are watching. Quickly go like the video as well, and if you go here, you can see the first 4 links are important. Enroll for 𝐂𝐤 NEET batch. Now, DNA batch, this is your DNA plus long NCERT series. If you click on the link and click on the view course plan, you will see the course plan. Buy the full course, click on that, it will load, okay. After that, you have saved 7,500 rupees. Click on C, 2,498, click on UPI, pay this amount and you will get the code. Clear? Very simple procedure, no breaking your head, no hidden GST or anything. You will get the scan, pay the amount and you will be able to finish this, okay. Clear to all of you? Yes or no? Okay, now same thing you can do with your other batches as well. You can just go, and if you want only DNA batch, just click on this and you can go here, view the course plan, here buy the course. Okay, here also the actual price you will be able to see. Okay, see 1499. Okay, again put UPI and you're done. Clear to all of you? So, at least for the books, I would request all of you, if you have not invested money anywhere and you are planning to buy any books, it's better you can enroll in a batch where you have lectures not only from us, from other teachers as well, and also you can access to all the lectures, be it JEE, be it NEET, all the lectures you will be able to access. So, why don't you just buy a batch itself, right? And also if you want just DNA batch and Tejas book, click on the link and you can see the prices here. Okay, right now it's again at 3000 rupees. Okay, Tejas book alone was that expense, but now it has become to this price. Okay, so it will go to 2998 rupees. Clear to all of you? I have shown everything. Later don't tell. Last time students are telling, ma'am, we did not see how to enroll, we are getting confused. Nothing to get confused, directly deviate from the link, put the UPI and you will be able to pay. Clear? So, make your decision wisely this Diwali and have a very safe and happy Diwali. Do let me know in the comments how you understood the entire immunity topic. So, it's not a small topic. We have done innate immunity, acquired immunity. We also understood about B cells, T cells, immunoglobulins, also about your vaccinations and your active and passive immunity and allergy. So, that major chunk of human health and diseases is done. AIDS, cancer and your malaria will be explained in the next class. Okay. So, I hope the session was really helpful. I really enjoyed the session because I could open Tattva book and ask you a lot of questions as well. I'm super duper happy with the session. I hope all of you also enjoyed. All these notes, okay, all the notes which are handwritten by me, I just came with blank slides. As you can see, I didn't even prepare one slide also. Everything I came and wrote right in front of you. So, all the slides will be available on the Telegram, okay. Thank you so much for joining. You can join my Telegram as well, Biology by Gopika, and also join Vedantu NEET English Telegram. See you all live in the next class. Happy Diwali till then. Have a safe and very prosperous Diwali this year, and I hope next year all of you will be able to celebrate your Diwali in your dream medical colleges and your government medical colleges. I hope and pray that most of my students are in AIIMS next year. So do your hard work, don't just depend only on the teacher. You should do your own self study. As I always tell, self study is very important, and practicing questions. As I told you, as soon as you finish this class, I want all of you to click on the NEET treasure box link, go to the link, solve the questions. Okay, solve the questions as well. I can show that also to most of the students who join new, because there are a lot of new students. Go down, join NEET English WhatsApp community. You can, below that you will see one link like this. Okay, now let's say I do not have any questions, ma'am, I do not have access to anything. See here, PYQs are put. So just click on this and you will be asked to enter the phone number if I am not wrong. Okay, yes, so you can start the test. Okay, you start the test, and if you click on that, your questions are already here. Okay, so these questions you cannot skip, you have to save next and go. So do this also. I want all of you to do this. Now I have taken photosynthesis in higher plants. You can go here, search for human health and diseases. So here is your human health and diseases. Okay, here if you go, click on this PYQ test, open, okay, start test, and your first question will come. Infection of Ascaris usually occurs by what? Drinking water containing eggs of Ascaris, eating imperfectly cooked food, ticey fly, mosquito bite. Okay, you select one answer and you go to save and next. Okay, again, which of the following shows coiled RNA strands and capsomeres? Like that, you can clearly do the questions. Okay, one by one questions you can do, and you can go and do save next. See, you cannot move until you select one question. Okay, one answer you have to select, then only you will be able to move forward. So I am clicking here, tobacco mosaic virus, save next and move forward. At which stage? Now every question will not be sure for you because you did not do HIV. Clear to all of you? I want all of you to go and practice without fail. No excuses, only practice. So thank you so much for joining, students. See you all in next class. Till then, tata, bye bye. Do not fail to comment and let me know how the class was. More such sessions will happen for all of you. Just let me know which chapter you want next. Thank you so much. Bye bye.